Today's CCB seminar on single cell data integration. Please join me in extending a warm welcome to our speaker, Ricard Ageluguet, who is a senior scientist at Altos Labs in Cambridge in the UK. And Ricard is a true expert and has developed several frequently used methods for single cell and multi-omics data integration. The most popular one is likely his multi-omics factor analysis, short MOFA framework, for which several extensions exist, such as MOFA Plus for multi-sample and multi-condition single cell data, and Mephisto for spatial and temporal data. So today, Ricard will speak a bit more generally about principles and challenges underlying single-cell data integration, and we'll also discuss applications to the integration of gene expression and chromatin accessibility data. So without further ado, I'm handing it over to you, Ricard. We are very much looking forward to the talk, and you maybe want to let people know how you want to handle it with questions, whether those should be done during the talk and the chat. I think we will also have some time in the end after your talk for some questions. Thank you. Over to you.
That's good. All right. First, thank you so much, Ludwig, for inviting me to present at the CCB seminar series. Actually, let me just share my screen now. Can you see it in presentation mode?
Yep. Okay.
Okay, brilliant. So again, it's really a pleasure to be here. Even if it's like online, I couldn't make it in person, but I may hopefully on the next time. And what I'm going to be talking today, actually, just if you want, you can find the slides here. But what I'm going to be talking about today is about computational principles and challenges when performing single cell like data integration. So this has been, the core topic of my research since I was a PhD student at the European Bioinformatics Institute with, under the supervision of John Marioni and Oliver Stegel, and then also in my postdoc stay at the group of Ulrich, and even now working as a senior research scientist at Altos Labs, it's still like the core area of like research, and even now I do find it very, very exciting. So. And I think, if you start diving into the single cell data integration field and you start like Googling and trying to find methods to perform data integration, something that you will realize is that there is many, many, many different methods. And if you read the title, or even if you read the abstract of those methods, they tend to be quite unspecific. And, all of these methods that are published, they rely under very different assumptions. The input data is very different, the methodology is different, and even the output is very different. And sometimes it's easy to get lost and confused just by reading abstracts and titles, especially if you have not followed the evolution of the single cell data integration field. So a couple of years ago, we tried to come together and try to come up with a classification of those methods that could make learning those methods a bit more a bit easier. And what we found is that, not surprisingly, the best way and the easiest way to classify those methods is by start looking at the input data. So at the input that those methods receive. And I think in general, any single cell or non-single cell genomics experiment that will generate omics data, you will end up with a series of matrices. So you're going to have groups of samples or groups of cells that are defined here as different columns in this case, and then you're going to have groups of features that we can call different views, different like data modalities, different omics, and so on. And then really the availability of those data matrices, which obviously depends on the experimental design and the data set that you're working with, is what will determine the type of data integration that you need to perform. So specifically, if we identified at least 3 major cases for data integration. And again, it really depends on which data matrices you want to integrate and what type of data you've got available. So the first one, assuming again features are here in the rows and cells or samples are here in the columns, is horizontal integration. So this is the case where you profile the same data modality across different groups of samples or across different groups of cells. Now, again, this could be an RNA expression experiment profiled in different institutes, different conditions, and so on. The second case, and actually what you can see here is that you have one dimension which is shared across your omics. In this case, is the features. So the genes are the same across your different data matrices. Now there's a second case that we call vertical integration, which is when those omics are derived from the same group of samples or from the same group of cells. So in this case, you have these matrices here from top to bottom. And importantly, what you see here is that now the dimension which is shared, the anchor between the different matrices, are actually the cells. So the features are allowed to be different. And then finally, the third case of data integration is diagonal data integration, which is the case where not the features and not the cells are matching across the different data matrices. This is obviously one of the more challenging data integration tasks because you don't have any anchors in the high dimensional space. So the way that people try to solve this diagonal integration problem is by either option one, you try and convert this diagonal integration problem into a simpler horizontal or vertical integration problem, or option two, you try to find anchors in a low dimensional space. So you try to infer low dimensional spaces for your different data matrices and then try to find anchors in this low dimensional space. So I'm going to give you some examples of the different data integration tasks. So the first one, and this is by far the most popular data integration task, is called horizontal integration, right? Which is, again, when you profile the same data modality across different groups of samples. And three very popular use cases, especially the first one, is batch effect correction, right? So essentially, you're assuming that every batch corresponds to a group of cells or a group of samples, and then you want to merge these data sets to remove the data-specific effect. So again, batch effect correction is by far the most popular case for horizontal data integration. Then there are other cases where, for example, in batch effect correction, what you normally assume is that your samples are independent, and again, you just want to merge them. But then sometimes you've got a hierarchy where one of your samples could act as an atlas, and then another group of samples can act as a reference. One group of samples act as a reference, the other one act as like query samples, right? So then you can assume this hierarchy to map those query cells into your reference cells. And this task is very, very, very common in the case of like single cell data. And then another example of this task, especially now that we're getting many single cell data sets across like cohorts of patients, is to essentially use the single cell representation of the data to learn properties and to learn representations at the sample level. So you want to learn something about the patients, something about the donors based on their single cell omics profile. And so initially, most methods for horizontal integration were batch effect correction. Then this second task became more popular. And finally, now we're starting to see methods that are dealing with this third type of horizontal integration. Again, just to cite some of those examples that perform horizontal integration, one of the first ones that was published in 2018, and it's still very much used nowadays, is called MNN. So it's based on matching mutual nearest neighbors between latent representations of cells. And then slightly later, there were some more sophisticated methods were published. For example, this is one of the most used one, SCVI, which is essentially a variational autoencoder where the batch variable is encoded in the input alongside your gene expression profiles. And again, both of these methods are very much used nowadays. Then it came this second wave of methods that allow you to map, to essentially take one of your data sets as a reference, the other data set as a query, and map them one on top of the other. And these methods, as I said, they came slightly later. So one of the ones that is very much used nowadays is this method called azimuth, which is derived by the Seurat folks. And then another one by Fabian Tha's group, which is called SCRChess. I'm not going to go too much in detail. It's just to give you some examples of those methods and which ones are still very much used now. The other type of data integration task which I mentioned is the vertical one. And this has been the main topic of my research during my PhD and my postdocs, and also now at Altos. Which again is this idea that when you profile all those omics across different groups, the different omics profiled from the same groups of cells. And this gives you a lot of power here, the fact that you profile those omics from the same group of samples. And one can find at least, two main types of analysis that can be performed when doing vertical integration. One is centered around the features, and the other one is centered around the cells. So for example, if you center the analysis on your cells, one of the most common problems is to again find a dimensionality reduction, try to find a latent embedding, but not now this latent embedding it uses information from all of your omics layers instead of just one of your omic layers. If you center your analysis on features, the easiest type of data integration you can perform, if you think about it, is just a Pearson correlation analysis. You can just take features from one data modality, features from another data modality, and then just run a Pearson correlation one against the other. This is the simplest task for vertical data integration. And just to remind you, this is really possible by the fact that you're deriving those omics from the same groups of cells. And then obviously you can do more sophisticated approaches to integrate this data and perform analysis on your features. And I'm going to talk about this. For example, inference of gene regulatory networks now is very, very popular in the single-cell multiomics field. So again, just to give you some examples of methods for vertical integration, one of the first ones that was published is the ones that we developed early 2018, the first version, and 2020. The second version, which was more scalable and more focused on single-cell data, which is this multi-omics factor analysis framework, where the main assumption is that this dimensionality reduction that you perform across all of your different omics is linear. So you end up with this matrix factorization scheme where you have some latent dimensions here that captures your latent representation of your samples, and then you have these data modality-specific weight matrices that provide you with interpretability of your dimensionality reduction. It allows you to tell which features are driving your embedding. And this is really, really useful when you care about the interpretation of your latent space. Since then, some methods have also been published, which essentially extended this using different frameworks. So one of the recent ones, which I like, is based on topic modeling, which is pretty good for binary data. So that's something that, for example, MOFA, it can handle binary data, but it's not really made for this. It works better with continuous data. Whereas since then, given that some of the data modalities for single-cell data are actually binary, like single-cell attack, for example, some people have developed models which are tailored to the specific assumptions and the specific needs for single-cell data. And this one, for example, is also linear, but since then, as I said, there has been extensions of frameworks like multi-VI, which is derived from the original SCVI, to handle multi-omics data in a nonlinear fashion. But there's always this trade-off. Obviously, when you use a nonlinear method, you have more explanatory power, but then you lose the interpretability of your embedding. And I'll stop in a second to ask some questions. Let me just get a couple of slides. And the main analysis that is centered along on the feature space is this inference of gene regulatory networks. And this is really popular, especially because most of the single-cell multi-omic essays are focused on profiling gene expression, like chromatin accessibility, DNA methylation, and perhaps also like surface proteins. So those four are the core modalities that are like mostly accessible that are essentially more accessible to scientists. And since then, there has been lots and lots of methods that have been developed to profile and to infer gene regulatory networks. One of my favorite ones is like Cell Oracle. I'm not a particularly big fan of how they build the gene regulatory networks, the assumptions that they make, but one of the real powers of Cell Oracle, and it was a beautiful work, is that they showed you how from those gene regulatory network models, and again, I'll show you an example of this later, how you can perform in silico like perturbations. You can in silico knock out TFs, and then it allows you to predict which genes are going to change and how the cell fate like decisions events are going to change by the knockout of a specific TF. Since then, there has been also other frameworks. One of the most popular is Cynic Plus, which it makes a slightly more sophisticated approach to build the gene regulatory network, but then it basically uses the framework of Cell Oracle to again predict the effect of like perturbations on transcription factors. And then one that actually came from Boston, from the group of Luca Pinello, it's called this Dictis method. That it's, I mean, the principle is quite similar on how they build the networks, but one of the smart choices that they've made here is that whereas those were really focused on, let's say, pseudo bulk estimates, so inferring gene regulatory networks either across the whole population or within specific cell types, in Dictis, they have formulated the method in such a way that you can model the changes in gene regulatory networks in a continuum fashion. So it's really useful when you want to study continuous processes like cell differentiation and so on. And for these, they use some smoothing approaches, and then they do some running windows where they infer and tune the GRNs in a dynamic fashion across some pseudotime. If you want to read more actually about GRNs, we wrote a review this year that has been published, and I'm just adding here the name and the reference. And then finally, before I take some questions, I just want to discuss the last type of data integration tasks, which is diagonal data integration. This is the most difficult case, as I said before, because here you don't have any anchors in the high dimensional space. The features are different across the assays, and the cells are also different across the assays. So this would correspond to, for example, doing single cell RNA-seq on this group of cells, single cell ATAC-seq in this group of cells, and let's say single cell DNA methylation on a separate groups of cells. So as I said before, the way that normally people try to address this problem is by converting it into a simpler data integration problem. So for example, let's say you've got single cell RNA-seq data, which is the one here, and then you've got single cell ATAC-seq data, which is here. Again, the features are different. So single cell ATAC data, you mostly quantify accessibility along peaks. So, accessible regions throughout the genome, which they do not necessarily match genes. Whereas gene expression is quantified on genes, right? So an approach to try and integrate these two data modalities is to essentially summarize and quantify the chromatin accessibility data on genes. So you would focus on like promoters, on transcription start sites, and then quantify accessibility along those regions. So in this way, you would essentially have the same features in one data modality and the same features in the other data modality. And then you turn these into a horizontal integration problem, and then you can use all of these methods that have been developed for batch correction and so on to perform the integration. Sorry. But then as I said, there has been some methods which actually have been able to deal with a natural diagonal integration problem by the fact that the features and cells are different across assays. One of my favorite ones is this method called glue, which again, you can see here the shape of the problem, which is very much what we have described. And then this is based on a generative model, very similar to the method called the CBI, which you make this like variational autoencoder. The main difference here is that they used a graph to essentially link the features from one modality to the other modality. So for example, in the case of single cell ATAC-seq data, you would link the peaks to the nearest genes, and you would encode these in a graph that defines your prior knowledge. This graph can also be derived from like public knowledge as well. You could take, pathways from ENCODE, or you could take some like publicly available resource to derive those graphs. And then what they've shown is that by using those graphs, you can get feature embeddings as well as cell embeddings, which are very, very meaningful. And actually, they show that the performance when you have this graph is much, much better than when you don't have this graph. So this kind of like introducing prior knowledge seems to be very, very important when you are dealing with a complex task of diagonal integration. And then finally, the last slide before taking some questions is that there are even more complex data integration tasks, such as the one, for example, that we call mosaic, data integration, which is when you have different subsets of anchors. So in some cases, you've got anchors that are features. In some cases, you have anchors which are the different cells. So you end up with this kind of mosaic. And then, to integrate this, you have to, again, deal with this fact that sometimes you're using features as anchors, sometimes you're using cells as anchors. But I'm not going to be describing methods in detail. There are 2 or 3 that have been developed for this specific task. Actually, this was supposed to be what I'm going to say here. This is the wrong link. I will fix this on the slides later. But if you want to read more about these different problems of single cell data integration, we wrote a review 2-3 years ago. It's actually not this one, so sorry, I will just fix this on the slides. But I'll share this later in case you want to read more in detail. So with this, for now, I'll take some questions on these basic principles and challenges of single cell data integration.
Maybe one question from me, Ricard. So thanks for this great structured categorization of these different methods. I think that is super helpful. Now, from a practical perspective, I often wonder, so how can we assess whether an integration kind of like worked well? Right, so many papers, like for example, if we take this batch integration or this batch correction problem, right? So many papers you see like these integrated data sets somehow presented on a reduced dimension plot, such as a UMAP, and then somehow the argument is being made if like data from different data sets merges well on the clusters, the integration worked well, but this, of course, like masks effects of, like over correcting or over integrating, right? Where you kind of like force certain cell populations together that actually do not belong together. So flatten somehow the biological variation there. So I wonder, are there, in the best case, some simple and intuitive metrics or diagnostics that one applies to kind of like quantitatively assess whether the integration worked or worked well?
I think the short answer is no. There are no easy metrics to try to assess this. And people have been trying many different types of benchmarks to try and compare those methods. So they try to quantify how much biological variability is like preserved and how much technical variability is removed, because that's essentially what you're trying to do when you integrate those methods. But this problem that you mentioned about sometimes like overcorrecting your biology, it's always a risk when you perform this type of data integration problems. My personal approach, the way I like to see it, is that I always first start with the simpler linear methods, and sometimes they are enough, and these they just work. Then in the second step is when I try to go for the more nonlinear methods. It's true that in the case of single cell data, I had little success using linear methods for batch effect like correction, because the fact that you've got all these different subpopulations, so there are interactions between the genes in the different subpopulations and so on, makes those linear methods much harder to work. But for example, if you try linear batch correction methods within a population, then in this case it typically works. So Lima and this type of methods, when you're trying to correct a specific populations of cells, it works. But if you're trying to correct the entire subpopulations of cells, all the different clusters and so on, then it becomes a lot more tricky. And in general, my advice is always, all of these corrections are simply wrong. That you are making assumptions which are probably not true, and we're just trying to get the best out of it. So think of it as you're doing something wrong, but sometimes you're gonna get something useful out of it. So if the correction that you get is useful for whatever biological question you want to answer, then just go for it. There is no easy way to say this method is better than this method, because sometimes method X works and sometimes method Y doesn't work, or it works. So I always advise try at least 2 or 3 methods. The one that gives you the most useful answer, then stick with it.
I have a question. So hi, Ricard. I learned about your method MOFA actually in the context of sample embedding, thinking of cell types as individual layers of an individual rather than like being the modalities they've used. And the way you talked about these methods, and for example, introducing dictus, which I wasn't familiar with, makes me think about how useful cell type adaptations are, but how in a way they're a restrictive view of the way that the single cell space behaves. And I wonder if you expect the future, or what do you expect the kind of the scene to look like in the future? Do you think that we'll do away with discrete characterizations of cells like cell types and move more in continua, be it through moving averages or parametrizations of graph space or something like that? Do you think that would be a useful direction for the field to go?
Yeah, I mean, I agree 100% with your point. And if you see about those methods that have been focused on GRN inference, is exactly the two cases that you mentioned. Either the first case, which they use discrete cell type labels, or the second case where they actually use single cell like trajectories, but quite simple ones, like 1 dimensions or like 2 dimensions instead. So GRN methods that are actually capable of inferring GRNs in a multi-dimensional space without the need of like cell clustering and so on. As far as I know, there are none of them. So I definitely see, a vacuum there that should be filled. And then there is the other aspect that some of the things that I see like coming up now very recently is approaches that, learning these GRN inference methods, there's a lot of data out there that could be used to essentially pre-train, let's say a foundational model, let's say that learns relationships between RNA and attack based on all the data from, let's say, ENCODE. And then you take this pre-trained model and then fine-tune it in your data sets. So these are examples which I think are very promising in the future, because again, people have shown in benchmark, and I think this is part of the review which I've mentioned, is that these GRNs tend to be very inaccurate. And that's because you simply don't have enough data to accurately train them and learn about all the context at which a specific transcription factor works. So, the more data you are able to fit into those models, especially using nonlinear approaches, the more powerful those methods will become. So this is where I also see a big gap that needs to be filled.
Thank you. That sounds very exciting.
Okay. So I'll take one more question and then I'll move on.
There's a raised hand.
Yeah. Hi, Ricardo. Thanks for the great overview. So I have one question about basically factorization methods. So I'm generally interested in interpreting functional interpretation of the results, and factorization methods are sometimes they are the only option available, but I'm also aware that the solutions that are provided by factorization methods are non-unique. How much do you think we can trust functional interpretations that we can get from feature decompositions and factorization methods, since you're the expert on the topic?
Can you be a bit more specific? So in terms of non-uniqueness, what do you exactly mean here?
So the non-uniqueness, it depends on the initialization or an assumption that you make, and the factors. I mean, basically the factors that you will get. My understanding is that they can vary quite differently, the solutions that you get to a data. But then, for example, in this example that you're highlighting here, if we go to each factor and look at what functions are represented in the dominant variables in each factor, we can perform all sorts of analysis, for example, enrichment, or see what functions these feature factor associations represent. But if the factors are non-unique in terms of that, using different initialization techniques, we get different factors, different assignment of features to factors. We may get different functional interpretations. So I hope that first my assumption is correct, but if that's the case, then how do we mitigate this situation?
Yeah, I mean, this is definitely a very good point. I would say, though, this is more of a point for nonlinear methods than for matrix factorization methods. In those cases, we know that the initialization makes a huge difference. Here, for example, in those optimization methods, like for example in MOFA, the landscape is almost convex. So then it's much easier to get, it's not 100% convex, like we don't have a proof that makes it convex, but by, just empirically, we see it to be like very, very much convex. So it's true that, with different initializations and different assumptions on, so for example, if you're using a Bayesian method, obviously the use of like different prior distributions will give you slightly different answers. That I agree 100%. Again, in my experience, I find this not to be such of a problem when dealing with the non-negative metric factorization methods or with MOFA and with other methods. This is more of a problem for nonlinear methods. But I agree with you, this is always super important to take care of, and always the advice when you run those methods, all these different hyperparameters that they provide, at least make sure that, some of the range is actually explored. And most people don't do this, especially even when running UMAPs and things which are very, very unstable by slight changes in the hyperparameters. People still don't do it. So I mean, I'm not sure if it's a good answer for what you're asking, but I do agree with the problem 100%.
No, thanks. It's very valuable to have your input on this.
Thanks. Okay, so I'll move on, otherwise I'm thinking we're running out of time. Right, so this is kind of like, I'll give you an overview on the theory of data integration, and then I will show you some practical use case. And this was, the work of my postdoc. Actually, this work started in my PhD days. And the idea of this project, this was in collaboration with the group of Wolfreich in the UK. We wanted to create a single cell multimodal atlas of mouse early embryonic development. So back in the old days when we started doing this, there was no commercial technology that allowed you to do single cell multiomics. So we had to come up with our own method. So here's where, together with Stephen Clark, and again, this is mostly his work, not mine, he developed one of these like technologies that allow you to profile epigenetics and transcriptomics from the same cell. So this method, I'm not going to describe it in too much detail because it's not very much used nowadays. I mean, we do use it a lot, but it has never become commercially available, and not many labs in the world have access to it. But this method is called SCNMT-seq. And again, I'm going to skip the details, but the main principle is that it works on plates, so 96 well plates, which implies that the high throughput is not as much as with the droplet-based methods like 10x and so on. But in each one of those cells, you get very good quality estimates for the gene expression, the DNA methylation, and the chromatin accessibility from the same cell. So it was actually the first method that allowed you to profile up to 3 data modalities from the same cell. And this was published, I think the method was published in 2017. And then we applied this method to profile the early embryonic development, and this was published in 2019. So this data set was really valuable back then. As you can see, it doesn't provide that many cells because it's a very expensive protocol to run. I think the price of each cell was around $70, so it was very, very expensive. And that it really limited us on the amount of cells that we could profile on, and how late we could go into early embryonic development. So we mostly focused on those stages from 4.5 to 7.5, which is where you start to see gastrulation and the emergence of the 3 germ layers. But again, the data was really useful to try and link all the changes that are happening in cell states that you can really measure in the gene expression to the changes that are happening at the epigenetic level. So you could find enhancers that are primed that determine specific cell fates and so on. But again, this was the work that we published a few years ago. But since then, the main revolution, I would say, that has come on the single cell multiomics field is the access to this 10x multiome. So the 10x multiome is co-commercialized by 10x Genomics, and it allows you to profile gene expression and chromatin accessibility, so essentially single cell attack-seq at very high throughput. So in each experiment, if the experiment works, because this kit is known to fail in a significant amount of times, but if the experiment works, you get around 5,000 or 6,000 cells per experiment, which obviously the throughput that you get is way higher than what we were used to get before. So essentially, this technology allowed us to go beyond the stages of 6.5 when gastrulation begins and try to dive into the states of early organogenesis, when you start to see the main organs being formed. So we profiled in this experiment 60,000 cells from the stages of 7.5 to the stages of 8.75, where the embryo really grows massively in size. So the first data integration task that we performed here is that, in this UMAP, This UMAP was actually obtained using MOFA. So you get the MOFA latent space by combining information from RNA and ATAC. Then you run UMAP on the MOFA factors, and then you essentially get this. And then you can see the differences in stage. But then to try and annotate cell types into this, rather than doing it de novo, which takes a lot of work, we follow this horizontal integration approach where we took one data set as a reference. So we had a highly curated single cell RNA-seq data set that was published a few years ago with all the cell types annotated. And then what we simply done here is to take the gene expression from our experiment and then map it onto the reference atlas. And you can see that this mapping, the 7.5 cells, were mapping in these places of the reference UMAP, whereas the 8.5 cells were mapping in the later development than in the like 7.5. So it provides a good QC that this mapping is working. So with just running one script, we're able to annotate all the different cell types in our experiment. And then, so that was using the gene expression data. So then from using the ATAC data, what you want to do is to learn gene regulatory elements. And essentially the ATAC data would look like this. So you would typically pseudo bulk per cell type, because single cell ATAC data is very sparse. So if you don't pseudo bulk, it's very, very hard to infer all these different ATAC peaks and gene regulatory elements. So here you would have each track here would be a different like cell type, and then you can see the accessibility. For example, around this gene in GATA6, you see that the transcription star site of the gene is constantly accessible across all the different cell types. But then you've got these distal regulatory elements which are highly variable and are very much cell type specific. So that was one of the main findings in our work, and many people have shown this as well, that in general, when you look at promoter regions, they are usually quite stable within these cell fate decisions. But really, what drives these early cell fate decisions is mostly enhancers. Enhancers are much more like dynamic than like promoters. But as I said before, the fact that you've got gene expression and chromatin accessibility from the same cell is really ideal to study gene regulation. So it allows you to query and to explore the function of like transcription factors and how they regulate downstream gene expression programs. Now, doing this, you typically need something. So you typically need information of which genes are being targeted by each like transcription factor. And the best way to know this is by typically profiling transcription factor binding sites using, for example, ChIP-seq data. Now, the problem is like ChIP-seq data is cell type specific, and you only have it for a few TFs. So there's no way that for a specific system you can get ChIP-seq data for all TFs in all of the different like cell types. If we had this, great, but we don't. So the alternative then, and that's what most people use, is the fact that many of these TFs, they have a sequence motif. So then you scan the DNA sequence within, let's say, within the ATAC data for those motifs. And then when you see a motif, you can assume that a transcription factor is potentially binding in this region. Now, there are two main challenges when doing this. The first one is that the number of motifs that you observe within a single ATAC within a single ATAC peak of, let's say, 300 base pairs or 400 base pairs is massive. So obviously it depends on the score and the threshold that you use to quantify motifs, but here we would typically get around 50 transcription factor motifs for every ATAC peak. So there is no way you can link a specific transcription factor to that specific ATAC peak just by using the DNA sequence motif. And then the second main problem is the redundancy of TFs from the same family. So the motif of TFs from the same family is typically very, very similar. And even though the gene expression patterns and the activities of all these different TFs is actually very, very different, they all share the same motif. And their activities is normally regulated by cofactors and binding partners that determine the specificity of those TFs. And that's something that you cannot do by just looking at the DNA sequence alone. So in this work, we try to extend these methods by trying to incorporate the fact that we know the gene expression for TFs. And that's something similar that other methods also do now, like Cynic Plus, for example, is also doing this type of approach. And we just call it, we just perform what we like to call it an in silico ChIP-seq, which was to try and use this information about, the chromatin accessibility, the DNA motif, but now incorporating the gene expression data from the TF to try and predict transcription factor like binding sites. So once we predict those transcription factor binding sites, we use this to predict what is the gene expression programs that are downstream of each of the TF. And briefly, again, the method here is very, very simple. It's just using effectively it's using the correlation between the gene expression of a specific TF and the accessibility of a peak that has the motif for that TF. So if there is a positive correlation between transcription factor 1 and peak 1 that has the motif for that TF, if this correlation is positive across cells, then this log here gives you the intensity, the strength of association between TF and peak. So you make an arrow. If there is no correlation, like for example, for transcription factor expression, 1 and this peak B, where you see no correlation, then the edge here is 0. So you do this for every combination of TF and peak. And then eventually what you end up is with a large network that links transcription factors to attack peaks. So, I mean, the first approach to see if this actually, this is very simple, that does this even work? So what we did, and mainly just focus here on this plot on the right, what we did is to fetch chip-seq data for TFs that we know are important for mouse early embryonic development, like CDX2, TAL1, GATA4, FOXA2, and so on. So what we did is compare the in silico binding score, so this strength of association that we see between the transcription factor expression and the peak accessibility, with the observed chip-seq signal for those TFs. And then we saw that the correlation was quite good. So it gives you some confidence that this method, at least for some TFs, is actually working. It's extremely simple, as I said, and it ignores complex interactions that we know exist between TFs. And we know that some enhancers only become active when you have cooperative interaction between TFs, but that's something that this model is not like, it doesn't take it into account. And actually, this is where I envision that the next wave of methods will come in, methods that are actually able to deal and learn about this TF like cooperativity and how by cooperating they regulate gene expression. But even with this simple method, we got something quite useful. And here are examples. So this is this TAL1 transcription factor, which we know is important for blood. And you can see how the accessibility, which is these tracks here, goes up in blood progenitors and then erythroids. And then when you plot the chip-seq data for TAL1, and then our in silico predictions for TAL1, you see that they actually match. So this was quite reassuring. And even for some TFs where the motif is quite unspecific, for example, CDX2, which is literally like this, and it's a very complex transcription factor, we see that, for example, in the Hox region of genes, where there is a lot of gene regulation, we're able to actually predict the binding of like CDX2. I mean, you obviously miss some peaks and so on, but obviously at the genome-wide level, the predictions are fairly good. And so this is the first approach. So essentially, we use this to try and link transcription factors to peaks. And then this is the first link. And then the second link is to try and link those peaks to the nearest gene. So that's how we would infer gene regulatory networks. So, in a pipeline, the way that this inference of GRN would work, and most methods do something similar, is first you define the cells that you want to use, because sometimes you don't want to use the whole cellular landscape. You want to focus on specific subpopulations of cells. And a specific advice I would give to everyone is that if you want to study a differentiation process or something continuous, do use meta cells. So I'm not sure if you're familiar with this concept of meta cells, but it's the idea that single cell data is very, very noisy, especially for epigenetics, especially for ATAC-seq data. But if you pool neighborhoods of cells which are closely correlated, then you pool the counts from those cells, and then you learn kind of like representative meta cells that capture your entire landscape. But then, the coverage of each meta cell is much higher than each single cell, and it really improves the GRN inference methods in my experience. The method I like is by Dana Perez's group, which is called C-cells, and I'll leave you the GitHub link in here. So that's the first step. You would select the meta cells of interest. Then in our case, we perform this in silico chip-seq to try and link transcription factors to attack peaks. In the step 3, you would link these, cis regulatory regions bound by TFs to the nearest genes. And then you would use something like 500 or 50 kbs, depending on how stringent you want to be. and then feed a second model that predicts the expression of a gene given the transcription factor that we predict is binding near that gene. And again, in the simplest setup, you can just use a Pearson correlation or a linear model, something similar. Other methods like Cynic Plus, they use nonlinear methods for this. But I'm quite comfortable using linear methods here. They tend to work quite well. But at the same time, nonlinear methods could give some powerful advantage. And eventually, that's what you end up with. You end up with a directed network where each transcription factor is linked to the genes that we make we predict that they regulate. So that's the principle. Is there questions about the GRN inference methods? I have this time for 1 or 2 questions here. Cool. Then I'll move on. So this is the approach that we followed, and I'm going to show you just briefly one example. I don't want to go too much in detail about the biology here, but we studied this differentiation process, which is called these neuromesodermal progenitors, NMPs, which are known to differentiate into either spinal cord in here, spinal cord cells, or into the somatic mesoderm cells in here. So again, taking all of these cells and applying this GRN inference pipeline, we're able to recover this complex gene regulatory network. And for simplicity here, I just made connections between TFs. So TFs to TFs. I ignored all the like downstream genes. And by doing this, we learned something useful. For example, this massive interconnectivity between the host genes, but then you learn some positive and some negative associations between TFs. Again, I don't want to go too much in detail about the specific of the associations. The thing I want to describe, though, is that one of the powerful things I see in these GRN inference methods is asking the following question. All of this data, which is derived from let's say a healthy wild-type mice, can this be used to predict the effect of knockouts? So just by taking a gene regulatory network inferred from wild-type data, can you predict the effect of knocking out a specific TF? So that's one of the main questions that we're interested in to answer. And again, the method that we use here is this really beautiful framework of Cell Oracle. That again, the idea is that you learn this gene regulatory network that connects transcription factors to genes, and then you take a specific node that you want to assess, and then you set the expression of this node to 0. And then what this framework does is essentially it makes a linear model that it propagates the signal through the nodes. And then by doing this, it predicts the changes that you see in the gene expression, which is downstream of the TFs. And then one of the really beautiful things about this method is that they came up with an approach to visualize the changes that you see in cell type identity using a similar framework from what people have done for velocity inferences. So you can see these arrows that for each cell, you can see if this cell is being pushed into this direction, or if this is being pushed into this direction, and so on. And you can do this systematically for every TF in your system. So this is the method that we applied, and actually we got very interesting results. So for example, applying this into the case of our neuromesodermal progenitors, what we found is that knocking out this gene brachyury, which is the one which is highlighted here, we can see that the arrows, typically those cells would either go, those cells here would go this way, those cells here would go this way. But now when we knock out in silico knockout brachyury, what we see is that the cells are being pushed into that direction here. Since instead of differentiating towards the somatic mesoderm on the right, the cells are being pushed and they differentiate into the spinal cord fate. And again, this is now going a bit too much detail into the biology, and I'm going to skip that part, but I'm going to keep it on the slides in case you are interested. But we did some experimental validation on this. So we performed an experimental knockout of this gene. We again profiled with 10x Multiome, and then we actually showed that the phenotype that we predict in silico is something that you also observe in the experiment. And then the nice thing about having gene expression and ataxic data is that you can then pinpoint exactly which regulatory elements are dysregulated by this specific gene. And then you can find some enhancers that prime for either one cell fate or that prime for the other cell fate. but because I think I'm running out of time, I'm going to skip this. But I'm going to keep the slides here in case you're interested. All right, so just to sum up, in this work, we have taken all these different, we have generated a single cell multimodal atlas of mouse early organogenesis by using this 10x multiome technology, which allows you to profile gene expression and chromatin accessibility from the same cell. Then what we did is to apply this algorithm to predict transcription factor binding events, and these transcription factor binding events, they were then used to predict gene regulatory networks, and then we link these gene regulatory networks to cell-fate transitions. And then finally, this is what I think is one of the highlights of this type of data and this type of methods. We use how computational models trained and applied on unperturbed data. It allows you to predict the effects of transcription factor perturbations. And again, we did some experimental validation of this, which I think, in my opinion, was actually quite nice to see that the predictions actually made sense. So with this, I'll just thank mainly the people that were involved into this project. And I thank you all for your attention. I'm happy to take any more questions.
Well, thank you so much, Ricard, for this really wonderful talk. It was super interesting. I had one more question with regard to the first part of your talk, if I may, on this mosaic integration piece. This seems to be something rather recent, and I find interesting. So when you have when you do integration on a set of partly overlapping features, all right, and there seems to be an application scenario here where you maybe have a whole transcriptome technology such as single-cell RNA-seq, and then maybe some of these new targeted spatial imaging technologies such as MERFISH or Xenium or COSMICS. And I was wondering, what's your intuition here, or what are challenges here? Are there any benchmarks how well this work? I assume you could do some sort of maybe cross-validation where you, on the features that do overlap, can kind of check how well you are able to kind of infer or predict those. Have people done that already? Do you know that?
Yeah, so you're exactly right. So that's actually the way that people benchmark this type of mosaic integration methods. As I mentioned, there are not that many out there. So one, for example, is called StabMap. There's another one which is called dictionary learning by the Seurat developers. But the way that those methods are benchmarked is by using, as you say, those assays that provide you with either horizontal integration or with vertical integration. Specifically, again, let's say you have single-cell RNA-seq data, single-cell attack-seq data, DNA methylation, but then you happen to have an assay where you have RNA and attack from the same cell. So this assay is typically used as a kind of as a left-out data set to try and then validate that. I mean, not the whole data set, but like a subset of this data is left out to try and validate that the integration is actually working. So, again, you need to make use of the fact that some of these data sets have anchors to try and guide your data integration approach. I have to say, I'm not really an expert on those methods. I've never developed one of these myself, so I'm just speaking from intuition here.
Great, thank you. Any other questions for Ricard?
Hi, that was a great talk. I have a question about, well, I'm curious if you advocate for additional supplementary experiments as an avenue to avoid some of the integration issues you flagged. So experiments conducted at different locations, you might knock in a known quantity of some well-characterized cell line, and that can help you characterize the batch effects. That said, I know that additional work is always a hard sell, so I'm just curious for your thoughts.
I mean, I think as a computational scientist, like most of you, I do agree 100%. So, if you can turn this diagonal integration problem into something that you've got some sort of anchor to play with, like turning it into some sort of mosaic integration, at least, it definitely helps a lot when it comes to benchmarking those methods. But then the main challenge is not doing the integration, is convincing the experimentalists to actually go and do the experiment. This is actually the main challenge.
Any other questions for Ricard?
like nitpick because I wasn't sure whether during the chipseek in silico, you don't seem to predict the actual clip signal or the shape or impute that. Is that just because you were using a simpler method, or is that a completely different problem that we're trying to solve?
Yeah, I mean, I guess this is a lot more of a complex problem. I mean, we did pick up to some extent when you take averages. Where is it? When you take averages, you see that the in silico binding score, so the strength of the correlation intuitively of between transcription factor expression and peak accessibility does correlate with intensity of the chipseek signal. So there's something in there, but I think this method would be too simple to dig into that. And I mean, something that we're planning to work on now, and I hope that other people work on this as well, is to try to extend this simple linear framework into something non-linear that takes into account the cooperativity between TFs and makes much more accurate like predictions of TF binding.
Fair enough.
All right, any other questions? If not, I would say thank you so much, Ricard, for a really great talk, and thank you for coming.
Yeah.
We will continue next week with, I think, a talk on protein crystallization, protein 3D structure. So if that is an interesting topic for you, please join us then. Other than that, thank you very much. Bye-bye.
Thanks, everyone. Bye-bye.
Thanks, Ricard.